Hello students. Welcome to my class Biology by Dr. Shabana Khan. We'll be discussing the Cambridge O-level biology syllabus. Now this is another video, part 3 of ATP Guidelines, in which we'll learn how to excel in tables and graphs. Now what we'll be covering is on tables and graphs would be parts of a table, and like different parts of it. What are the different types of tables, different parts of a table, the concept of variables, and a few of the past questions. We'll be looking at some simple tables and some of the more complex tables. Now in the graphs, we'll be looking at different types of graphs and discuss each type in detail with past paper questions. And let's start with our tables. Now let's talk about table. Now for example, what is a table? Now what do I want to find out? For example, I want to find out the height of some students in my class, 4 of them, and at the end of the investigation, after measuring the height, what do I get? I write down the height, all the names of the students and the height, and then what do I see is because that will not be enough, what do I do? Just 1 minute. So what do I do? I'm going to put down the names and the height of the students in columns and rows like this. So this is a table. And what is a table? A table is actually a graphic organizer used in scientific experiments and elsewhere to collect and organize data in a neat and clean manner, so it's easy for someone to observe. Now what are the parts of a standard, you might say, a standard data table would be, for example, this is a box with different rows and columns. You all know that, right? Now whenever you get a graph, like whenever you get a table like this in exam, remember you have to draw this with a pencil and a scale. So don't draw a freehand diagram, make it as neat as you can. Now the rows and the columns, let's see. So the columns are the ones and the rows are on the vertical and the horizontal ones. So columns are the vertical ones, rows are the horizontal ones, and then at one side here we can see is going to be called the body of the table. Now so at the top we can see is the header or the main title. We need that is going to be the subtitle. Now after that, we can have separate headings for the first column and the rest of the columns, okay, depending on we'll see what is going to be the experiment. Now the data is written, all right. So this first column, we'll see what comes where and why have we kept them separate. Now whatever numbers, whatever result, whatever data I'm given, I'm going to put them down in these spaces here, and each of them is termed as a cell, right? And this forms the body of the table. Now the main point that we need to understand right now is that the units are never, of the numbers are never written in the body of the table. They will always come in the headers, and so no unit in the cell or the body of the table. And if you do that, well, you're going to lose 1 mark straight away. 1 mark is lost. All right. Now for example, now before we move further, we need to understand the concept of variables. All right. So variables by definition are factors which we can change or test, cause and effect in an experiment, but it simply means those things that we manipulate or observe in an experiment. Okay, what we handle, temperature, concentration, volume, time, and all of those things are all collectively known as variables. Now so variables are going to be of 3 types, right? Independent, dependent, and the constant variable. Now what does each stand for? So first of all is the independent variable. Yeah, the independent variable, like I've labeled I here and I. So this is the thing that I will adjust at the beginning. Whatever experiment I'm going to test on, for example, I want to see the effect of temperature on the rate of a reaction. So the effect of temperature, so that means I have to work out the temperatures. I'll be using a number of temperatures and I'll see the result at the end. So this is what I will be adjusting at the beginning of the experiment. Hence it is termed as the independent variable, what I change or what I manipulate at the beginning. All right. Now what comes next is going to be the dependent variable, right? And the dependent variable, remember, is going to be our result. Yeah, and the thing or the data I get, which I observe, or which the numbers that I get at the end of the experiment, that is going to be my result, and that is known as the dependent variable. And why it's called dependent? Because that will be dependent on the independent value which I had adjusted. So just make it easy, don't make it too complex. Now, the last thing is going to be our constant variable. Now, as the name suggests, is something which I need to keep stable or constant. So this is pre-planned. Before we start the experiment, like along with the independent variable, we have to work out the constant variable. This is done before we start the experiment, all right? And because, see, we know that we're going to repeat the experiment at least 5 times for each variable. So we need to have a more reliable result, right? So how do I make sure that each time that I do the experiment, everything is kept the same except for the thing that I'm testing for? So what do we do? So there are a number of things, many things, and we'll be keeping them constant. And these are known as, so this is pre-planned and before the experiment, constant variables, right? Now the variables in experiment now, independent, dependent, constant, what I change, the result, and what I keep the same. Now what comes where? Well, let's have a look now. Independent variable and the dependent variables. So whenever we handling tables and graphs, we can remember a mnemonic for both of them, right? And independent, the I is important, what I adjust or decide. So I can make a mnemonic called fix. And you can work out mnemonics for so many things. Like we used to work in the medical colleges when we had to learn a number of things. So what we used to make is freehand mnemonics, and you can make some effort and you can make mnemonics for so many things on your own. Now I stands for independent, yeah, and the F would stand for, no, excuse my handwriting. So this would be, if I'm working on a table, so this would be the first column, all right, in a table. Generally in biology, unless specified, most of the time the independent variable will be plotted on the first column in the table. Now, if I'm working on a graph, so the independent variable or the first column will come on the x-axis. All right, I hope you understand. What about dependent variable? Well, dependent variable is going to be my result. So I'm going to use this R. You can make any mnemonic that you want, right? So I'm going to make a simple mnemonic like dry. Right? Now dry, dr stands for the result, and d obviously stands for dependent, and y would mean, so if this is the first column in the table, so that means the other columns, it's understood that in the table the other columns that come on the, you might say on the right side, it could be the result, and you can also say the right side of the table of the first column will be dependent. And y, if I'm again going to plot a graph, so this would come on my y-axis. Okay, so moving on, what comes where? For example, the previous experiment that we had done, right? The independent variable was, I had chosen a few students, right? So I'll write down the name of the students, and that becomes my first column. And according to fix, this is my independent variable, yeah, and that will come as a first column, and height and all, so that becomes my dependent variable, yeah. So independent comes here and dependent comes here according to the mnemonic dry. So this is going to be the rest of the table. Now let's have a look at a question. Now one of these tables, now this is going to be a very simple table. Now one of the questions that came in the year February, March, this is the code for IGCSE, if you know how to read this, February, March series of exams in the year 2023. So this is the year, this is the series, this is the CIE code for the paper, which is IGCSE, Biology IGCSE, and this is variant paper 6 and 2. So we have variant 1, we have variant 2. So we don't say 62, we can easily say 62. And this is for question number 2. Now look at the question. Scientists wanted to find out if drinking a certain juice lowers the blood pressure. So you want to find out the effect of one variable on another variable. Now first thing that you have to think of, which is the independent here, now which is the dependent here, and what can I keep constant. I'm not going to go into too much detail because this is a very simple table. I need to move on to the other questions. So what did you do? You do 2 groups of men had the systolic blood pressure measured. Okay, so two groups of men. Now we selected two groups, group number 1 and group number 2. So this being what we select, what I selected, so this becomes my independent variable, right? So this is my independent variable, and there are two groups, group 1 and group 2, and this should come according to fix in the first column. And this is exactly what I've done, is that I've placed a group number 1 and group number 2 in the first. So these are, this is the table here. So these are the headers, and this could be a subheader or subtitle, yeah, and this is the body of the table. I think it's, and these, each of these are cells, yeah. And so the group 1 and 2, this is my independent variable, comes in the first column. And whereas now what did I do? Group 1 drank two different kinds of, we have two different juices now coming in. So what did I measure? What was the result? What was the, what do we say, what was the outcome? What did I have to measure? What was the result? That is going, what did I observe was the blood pressure. And what pressure? Systolic blood pressure, right? So then we took two groups of men, and we haven't mentioned how many men in each group, but we took out a mean. So the mean systolic group in group 1, right, and mean systolic blood pressure, and this is the unit, millimeters of Hg is a unit. Now group 1 drank 500 cm cube of beetroot juice, yeah, so beetroot juice would be here before drinking. And so group 1 drank the juice, so you don't have to mention it in the table. And group number 2 drank apple juice. So two different juices, and we're looking at the effect on the blood pressure. So because we want to find out whether this juice really affects the blood pressure or not, right? So group 1 and group 2 are here on the first column. That becomes my independent variable. Now both of them drank the same amount of liquid, all right, and that was 500. That becomes my constant variable, yeah. And then what did I measure? I measured the mean systolic blood pressure, right? After 6 hours before, and this is after 6 hours, we measured the systolic blood pressure, was measured again. So before drinking, this was the blood pressure, and you can see it's almost the same in both the groups. And the group 1 drank beetroot juice, whereas group 2 drank apple juice. Just for your ease, I'm just writing it down. So before drinking, the blood pressure was this much, and now after 6 hours, the blood pressure changed to this, and then it changed to this. So this is the mean systolic, right? So this was a simple table. This was given in the question, but this is a very good example of drawing a very simple table. You have two variables. One is going to be the type of juice, and we have two groups because each group will drink a different group juice, and then we have the mean systolic blood pressure, and that becomes my dependent. So this is my independent, and this becomes my dependent part of the table. And because it was before and after, so I had to make a subheading or a subtitle, before drinking and after 6 hours, and I put the numbers here. Now if you can see, there are no units given here, right? So none of the cells have units here. I've placed the unit right at the top, and you do not have to repeat the thing. It goes for all of the cells, all right? This is example of a simple one. And you got some information as well out. And what was that? All of them were men, yeah. So they were of the same gender, and then they drank the same 500 cm cube of both of the juices. The volume was the same, so the volume of the juice was kept the same. All right, and we can measure, yeah, after 6 hours again. So what was kept constant for both the groups was the time. The time duration, you might say, was the same, and that was 6 hours. So one, from a couple of lines, we had extracted the independent variable, the dependent variable, that was the blood pressure, so in short BP, and the constant variable. And from just a couple of few lines, you can get so much information, right? So I hope you find this useful. Now let's move on to our student investigator. Now May, June 2023, 0610, and then 61, and yeah, the student investigated the effect of temperature on the rate of respiration in yeast cells. Okay, now on the effect of, now let's just read this: effect of temperature on the rate of respiration in the yeast cell. Now, what's a yeast cell? Have a look at this, right? Yeast cell, unicellular organisms, they are eukaryotes. And how do we differentiate them from the others? Usually we will see a small bud arising from the surface. We see a small bud arising because this is how they reproduce by asexual reproduction in a process known as budding. So yeah, so effect of temperature, will that be my independent variable or the dependent variable? What will I keep constant? Yeah, so let's have a look now. So yeast, yeah, when you give yeast, if you give it sugar, it will do what we say anaerobic respiration or fermentation. And what does it do? It is going to no, I'll just stop here. So it does alcoholic fermentation, and for that it releases energy from the sugar source, but in the process it's going to make two byproducts. One of them is ethanol. All right, so that's why this reaction is termed as alcoholic fermentation, and then the other will be the gas. Now, same equation we've used in biotechnology in making alcoholic beverages as well as in bread industry right now. So we can use this same reaction, because if you have a gas being formed, it's very easy to observe it in the lab. Yeah, so let's see how we can do this. And we want to see the effect of temperature, whether increasing or decreasing the temperature, will it affect the rate of production of CO2? What apparatus can I use? We'll have a look at this. Now, don't get perplexed by what's here. Now, so you want to see the effect of temperature. Now, what do I see? What do I see? Now, first of all, I'm going to keep the living sample in a test tube because these are microorganisms. So they're very easy to display and demonstrate in the lab. A small test tube will do. So I'm going to take the yeast and I'm going to keep it in a sugar solution. That will be the source of energy for it. The yeast in a sugar solution and vegetable oil, so that none of the oxygen gets inside, and because we want to keep it anaerobic, without oxygen. So the yeast is there with the sugar, and it will feed and will start to produce the gas carbon dioxide. The carbon dioxide will arise from here. Any gas occupies volume, it needs to expand, and as its production increases, what will we see is that we can't see that CO2 is a colorless, odorless gas, tasteless gas, but the gas will seem to move from here, and after a few minutes what we will see, bubbles will arise, and we are assuming it's carbon dioxide because lime water will turn milky as well. So lime water is an indicator for the presence of carbon dioxide gas because it will turn milky. Why is a bung there? To prevent any escape of any gases going in and out. Now, to further the experiment, I need to have different setups at different temperatures, right? So I've kept a thermometer in a water bath, and I can vary the temperature of the water bath and see the effect of production of carbon dioxide. Now, I'm just going to cut this short, and then we're going to move on. Oops. Yeah. So this was here. Yeah. Now, so what was the question? Now we have to prepare a table for the results and record the temperatures shown in two figures that are already given. Remember, you're not doing it in the lab. It's a theoretical paper, so you can't see the thermometer. So what the examiner has given you, they've given you this. Yeah. So they've given you two figures, figure 1.2 and figure 1.3. So they've done the experiment for you. Now you are given a certain amount of data, certain numbers, and then what do you do with them, right? So what was the thing? Prepare a table. You have to prepare a table, right? So move on. So now the first figure 1.2 is shows two thermometers, right? And you have to take the reading. So read the scale units, right? The numbers, the lines that you see are called units or scale units of the mercury column, right? And then figure 1.3, the other figure, this is showing the data collected by the student with hot water and cold water, how many bubbles and the number of bubbles. Now hot water, what do you think the temperature is? From 0 to 10, they've given 5 lines only. So that means each small number will represent two numbers, right? So if it ends over here, so that means this is going to be 41, 42 here. So the hot temperature was how much? 42, and hot water was this, and the cold water, it is less than 20 by one small digit. So 20 minus 2, that makes 18. So 10, 12, 14, 16 and 18. So the cold water, the temperature was 18 degrees Celsius, right? So prepare a table for this. So we have this. Okay, now the table would look like this. What do I have in my hand? I have the temperature and I have the number of bubbles. It's a very simple table. So we have two variables. The first one is the temperature that we had adjusted, because I'm going to change the temperature of the cylinder, the measuring cylinder, using a water bath. And the number of bubbles I'm going to count in the end. So what is what? So which is the independent, which is the dependent? So obviously the one that I adjusted, this is my independent variable, and the one that I measured as a result at the end, now this becomes my dependent variable. Okay, so the temperature of the measuring cylinder, and we know what that is, and then the number of bubbles counted in 3 minutes, but that, because that was given in the question. So be very, very precise, very specific, So the final table will look like this. Okay, you fill in the data and this is what you get. And mind you, no units are given here. Number of bubbles, we just have to count them, the unit needed. The degree Celsius, this is given here in the top. This is the title, yeah, the header, and this is the body of the table. Yeah. Okay, now let's have a look. This was a very simple table. You got 2 columns only. Now let's have a look at another experiment. This came in the year June 23, May-June series 2023, and the paper is the second variant of paper 6, question number 1. You can read up the question because I don't want to go into the details about the question, but let's have a look. Now the data that was given was, this question was about homeostasis, right? How much can we lose heat? All right, if this is the concept was of huddling of animals when they're all together. So why do they huddle? Does that help them to lose heat a little slower? So are they able to retain that much heat? Does it keep the body warm? Right, so we're going to experiment with 3 different apparatus. So this is like with 1 test tube, and the second one was with 3 test tubes together, and they're all held together by a rubber band. And the 7 test tube was this one. There's one in the center, where the 6 of them are surrounding this one. The 3 of them are here, like this, right? So now what I have to check is how fast do they lose heat. Okay, so all these tests were filled with all of these were tested with hot water, and then we recorded the time it took them to lose their heat, and we will record their temperature. So prepare a table and record the results shown in figure 1.5 and your answer to whatever you have to read from the thermometer, the first one. So what do I have to what's in my hand is the time. I'm going to start the stopwatch, and I'm going to measure the temperature after every 1 minute. What do I adjust? I will adjust the time, because I will have the stopwatch in my hand. I can stop, or I can just observe. After 1 minute, I'm going to look at the temperature thermometer again and again, right? We keep the thermometer in the test tubes, by the way, right? So what do so 1 test tube and 3 test tube and 7 test tubes, what will I do? So there are 3 different experimental setups, and the timings are after each after every 1 minute. That's in my hand. I am going to see the result. What will I observe will be the change in temperature. So if it's 1 test tube, after 1 minute it was this, after 2 it was this, and degrees Celsius, right? Now when there are 3 test tubes, in the beginning it was this, then after every 1 minute I have to record this. After 7 tests, so this data is already given to you. You have to put the numbers in a table. how many columns, how many rows do I need, right? So let's have a look. So the table, approximately it comes down to this. Now first of all, I have the temperature to think of, the temperature in the 3 test tubes, right? And yeah, and then there are 3 experimental setups, so 1 test tube, 3 and 7 test tubes, right? So what can I temperature of the test tubes, and the unit is degrees Celsius, and then I'm going to add 1 test tube, 3 test tubes and 7 test tubes. There's no mean calculation given, otherwise I would have made a column on the right, added a column on the right side. Now what do I do? Now comes the time. The time is in my hand, so I'm going to take it up as the independent variable. Yeah, so time in what, and the units are given in minutes, so time in minutes, so at 1 minute, at 2 minutes, 3, 4 and 5, right? Now what do I put in the cells? What I'm going to put is, against each of the test tubes, I'm going to put the numbers that are given. 65, 61, 58 and 55. The first was supposed to be measured from an image. Now in the same, in the third and in the seventh. So what do we get? Finally we get a table like this. Okay, so I hope you understand. Sometimes, yes, the tables are not easy, sometimes they are complicated, but the more you practice the past papers, my dears, the more you'll become comfortable with them. All right, so do make some effort. The more papers you practice, you still have a couple of days. Yeah, okay. All right. I hope you learned something, and thank you for watching, and do leave some information in the comments, any comments that you feel was missing, or if you any other topics that you need to be covered. All right, so watch out for other videos, and again, like, subscribe and share. Thank you so much for watching again.